shape of your houseplants by doing the pinching off of the apical meristem. And so if you do that, this computer, there we go. You can actually change the overall shape of the shoots. So here's a picture on the left of a plant that's growing normally. The apical meristem is actually out of the image. It's up there. But what they're trying to zoom in here is the axillary buds. So you have these axillary buds in the node region of the stem that you can see, especially that one that I circled, the very first one that I circled, is dormant. It's not doing anything. It's just sitting there. It's not growing into a new leaf or a branch or anything. And that's because of apical dominance. So when we say a plant is exhibiting apical dominance, what you look for are those axillary buds. And if they just look dormant like that, they're not growing out into anything, then the auxin in the very tip of the shoot, what happens is the auxin's getting produced in the tip and it's flowing, the auxin is flowing down the stem tissue and it's telling all those axillary buds, be quiet, don't do anything. You're good, just go to sleep. All right. And then if you take the axillary apex, the shoot apical meristem off. So here you can see in this lower right image, they've amputated the main branch quite severely. And now you can see that the axillary buds have sprouted and become active. So what has happened there is you've removed apical dominance by cutting off the shoot tip. No more auxins flowing down there to tell those axillary buds to be quiet. And now they sprout and start growing new leaves or branches. So depending on the plant, when you do this, you can get a bushier plant instead of a tall and leggy one. You'll get one that's more bushy. So I do this all the time with plants in my home. If I want that more short, squat, more full look to it. So they're trying to show that with the apical dominance, the auxin is slowing down the stem and keeping it quiet.
You just said like it flows on the plant, but is there a specific route that the hormones will take, like the phloem or?
Yes, yeah. So auxin can go from cell to cell to cell to cell, which isn't terribly efficient, right? But it can do that. But then it can also go through the vascular tissue for long distance transport. And we'll find most hormones do that. They'll use the vascular tissue, the xylem or the phloem, in order to move long distances. So, yep. Okay. So that's something simple that you should keep in mind if you ever have houseplants that are getting a little too long. We also see something that you may have done already if you've ever taken a cutting of a plant, if you've been at a friend's house and like, oh, I like that plant. Can I take a cutting of it? And so you take a cutting and then you put the cut end into maybe a little bit of water and you wait for the roots to form. Those roots technically are called adventitious roots. Adventitious roots. So that's what you're seeing in that top picture where someone took a cutting and put it into some water. And you see now that there's roots forming right where the cut was made on the stem. So what's really happening is pretty awesome. You have this stem that's been cut and injured, and now all those cells right at the cut area are like, oh, I've lost my root system. I need to regrow some roots. And so the stem cells, those stem tissue, have to totally morph and say, I can no longer be a part of the stem. I have to become roots. And so they'll shift a lot of their gene expression and start forming roots instead. So it's really a pretty amazing process going on. You just can't see it with the naked eye until the actual adventitious roots start to come out. And so here you also see adventitious roots on a vining stem here. And they're using that. It's probably really an ivy plant. They're probably using those adventitious roots to maybe attach to another plant or attach to a brick wall, something like that. And so anytime you take a cutting, if you've ever bought, like, I think it's called root tone or rooting hormone, they'll call it, or it can be a powder or a gel. and you buy it and they say, use this to dip your cut stems into. It's because it has auxin in it, and that powder has auxin in it. And so you can do that and it will speed up the adventitious root formation, but you don't have to use that. Don't feel like you need to spend money on that because the plant itself will have auxin in it. So the auxin will flow down to that area and say, make roots, make roots, make roots. And so you don't have to necessarily buy that stuff. Here you can see some adventitious roots on an orchid plant as well. So the auxin hormones are very famous for that. And then another way that we use auxins in industry is to do cloning. When we clone plants and do tissue culture production of plants, you have to supply some auxins into the growth medium that you're growing the cut plant on. And when you do that, it will tell the plant's cells to just grow, grow, divide, grow, and grow. And when that happens, initially, if auxin is the only hormone that you put into that growth medium, initially what you'll see is just this mass of cells. And we call it a callus. That's actually how they refer to it. And so if someone says, oh, look at that callus, they're referring to just a clump of plant cells resulting from lots of growth, but they haven't they haven't differentiated, like they haven't become root or leaves or stem yet. They're just a bunch of plant cells. And then if you take some of that callus tissue and put it into a new growth media with a different hormone, then you can get that callus to start differentiating and growing into a little baby plant. And it'll be genetically identical to the plant you started with. So cloning plants is big business. They do that. Most of the African violet plants that you see for sale, for example, in stores, are all done through tissue culture. And many other house plants are done that way. And then it's also used a lot in research because cloning, getting the same genetic material to do experiments on is often desired. So stimulating cell growth in tissue culture is a big deal. We always apply auxins into the recipe. So tissue culture is the idea of cloning plants. And then phototropism and gravitropism. We're going to find that we see auxins involved in that too. So we've already really talked about phototropism when we looked at Charles Darwin's research just a few slides ago, and we saw how the plant was bending toward unidirectional light. So that is phototropism. Photo is referring to light. Tropism is a growth pattern. And so what's happening is I keep saying it's bending toward light, but a more accurate way would be to say that it's growing toward light. And let me see if I have where's my picture? So here is an image of it growing toward light. And what happens is when you shine unidirectional light onto the stem, the auxin flows down. It starts being produced in the shoot tip, and then it shifts over to the unlit side or the dark side of the stem and starts flowing down the dark side, not the lit side. And so that blow up is showing the auxin is tagged with a blue dye. And you can see that it's only really flowing down the unlit side or the dark side. And anytime I think or say dark side, I think about Darth Vader because I used to watch those movies all the time when my boys were little and they'd watch it. And so, that took a lot of like decades of research to figure this out, but they finally did figure out that when you shine unidirectional light on the plant, auxin will flow down the dark side and it stimulates the dark side of the stem to grow. So those cells are growing, 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 growing. The lit side is not growing, and that's how you get that bending pattern. So it's really a growth response toward the light. Now, if you turn your plant, 180 degrees away, it will bend back the other way. So it's this continual growth response. It's adaptable. Wherever the light's coming from, it'll start growing in that direction. Okay. So that's why it's okay if you want your plant to grow more upright, you just keep shifting your plant in the windowsill. Okay. So let's see. Gravitropism, can you think about what that might be in response to? At least they named it something that's decipherable. So gravity, right? It's gravity. At least it's something that's easy to say and actually named after gravity. So we'll talk about that too. Another group of hormones are the gibberellins. Again, there's a lot of different subtypes of gibberellins. We often refer to them as gibberellic acid, so they're often abbreviated as GA, gibberellic acid. And this picture is showing its most famous attribute, which is if you spray stem tissue and leaf tissue with a dilute solution of gibberellic acid on dwarf plants. So these are dwarf cabbage plants. Those are the controls that didn't get sprayed below the ladder there. And then you can see these plants got sprayed with gibberellic acid, and you can see this huge increase in stem growth. So that's their claim to fame. So they really stimulate stem growth. So you can imagine if you are a grower and you're growing sugarcane for a living, it's the stem that you want to harvest for the sugar production. And so you can bet that growers do spray the gibberellic acid onto their plants for good sugarcane. The yield is tremendous when you do that compared to if you don't spray. Here's another not quite as dramatic example of, in this case, these are dwarf pea plants. So we do this in Plant Fizz, where we start with dwarf pea plants. Those did not get sprayed on the right. The ones on the left got sprayed with a dilute gibberellic acid solution. And so when they say it's a dwarf plant, it just means that it's producing gibberellic acid, but at such lower levels than normal that it really doesn't do much to the stem growth. And so if you spray it, it can still respond to gibberellic acid. It's just the dwarf plants aren't themselves making a ton of gibberellic acid. Here's a really good example too, if you're a wine grower or grape grower for wine production or just table grapes, spraying the grapevine with gibberellic acid, what it actually does is it spaces out where the grapes form on the vine. And so it gives more physical room for the grapes to grow bigger, sweeter. And so again, if you are a grape producer, that might be a trick that you would want to do. And so the ones on the left here did not get sprayed. That vine did not get sprayed, and you can see the grapes are more compact and clustered together. They're smaller and not as sweet. And so we tend to like bigger, sweeter fruit. And so the ones on the right were the ones that got sprayed. And so there's a lot of use of these different hormones. Once they figured out, what do these hormones do for the plant? And then when you think about all the people who grow for food production, they're taking advantage of this if they can to better the crop yield. Beautiful images here of hopefully what your seeds are doing right now in the dark or under the grow lights, I guess, in your sandwich bags right now in the wet paper towel. Hopefully they're doing this. And so gibberellins are important to get a seed germinating. So the seed itself has to produce a fair bit of gibberellins in order for the seed to fully germinate and start growing into the seedling stage. Do you remember from lab, or we talked about this maybe last week? What's that structure coming out of the seed?
The radical.
The radicle. All right, I do need you to know that term. The radicle is the embryonic root. And again, it makes sense if you know a little bit about seed anatomy, most seeds have enough fats in there and carbohydrates in there to sustain the seedling for a little bit, but it doesn't have water. So part of the process of getting a seed to fully mature is it goes through dehydration, because you wouldn't want to be a lovely plump water-filled seeds going dormant. Fungi would love it and they would attack and kill that seed for food. And so you want to dehydrate, go dormant. When gibberellic acid gets activated, it pretty much triggers the radicle to get growing and get out there and get access to water in the soil. And then hopefully the rest of the endosperm or the cotyledons can feed the baby seedling until it can start doing photosynthesis. So gibberellins are known to promote seed germination. In the beer industry, they actually use this. At least some beer companies will do this because hops, or I'm sorry, barley, you want to get barley germinating and breaking down the starches in the seed into simple sugars so the yeast can start fermenting it. And so they'll actually put in a dose of gibberellins to start that germination to try to get the starch broken down so the yeast can have an easier time. They also know that gibberellins are important when the plant has to go into the next phase from, so we don't really say puberty in the plant world, but they do have something very analogous to puberty in the sense that, you know, you have a baby seedling that's growing and initially it's very young and then it'll get bigger and stronger and but it's not able to produce any sperm or egg yet. And so it's the juvenile phase. We don't call it puberty. We say it's the juvenile phase. So if someone says, oh, that's the juvenile phase, the plant's still in the juvenile phase, that means that it's not able to produce flowers or sperm production, pollen. And so getting that shift from juvenile into the adult phase, we know that gibberellins are very important in that process, which is hugely important for the plant because, of course, the plant wants to be able to produce seeds into the next generation, and you need sperm and egg to get that to happen. So a little bit about gibberellins. Another group are the cytokinins. I put this up here. If you took BI 111 recently, that should look vaguely familiar. That's the mitosis process. And they know that cytokinins regulate that quite nicely. And you have to have the right dosage of cytokinins to allow for mitosis to occur. So mitosis basically is just, you know, getting its cell growth, right? You get the idea of the genetic material copying itself and then splitting into two equal halves, and then you get two cells resulting from that. So they regulate that whole process. That is an extremely important process for any living organism and plants. They also know that cytokinins are commonly produced in the root tips. That's one of the favorite spots for production. But you can also find it in really active young leaf growth, fruits and embryos. And in fact, they can't really make a plant that's a mutant for either auxins or cytokinins. Like they've tried to do that. Like I just want to make a plant that can't make auxin and it can't make cytokinin. And it might germinate, but it really it like aborts. It stops growth and then it dies. And so auxins and cytokinins are so important and so fundamental for the plant that they still can't create a mutant that doesn't have those hormones. The plant just doesn't make it. So it can suppress what the axillary buds or the lateral buds. Remember, we said auxins are suppressing that too. If the auxins are in the shoot tip and they come down the stem, they will suppress that. Well, the cytokinins can also do that. And then they've also found through quite a bit of research that in order for chloroplasts to be fully developed and fully functioning, you need cytokinins. And again, that's such a fundamental part of the plant. If plants don't have good, mature, functioning chloroplasts, they're not going to survive very long because that's where all the photosynthesis is taking place. That picture, of course, right there, that's the root tip. You have a really nice picture. So the root apical meristem is in this region, and then all of this region is the root cap. And then these images are trying to show what's called senescence. And senescence, that's just a fancy way to say normal cell and tissue death. And so every fall around here, our deciduous trees go through senescence. It's programmed cell death. And so, you know, we get the advantage of seeing the lovely fall colors as a result of that. But basically, when you see the fall foliage, what's happening is the leaves are going through senescence. And so cytokinins can actually delay that. So as a grower, if you're trying to grow certain plants and you want to extend the growing period, you can actually spray your plants with dilute cytokinins and it slows down that programmed cell death. It's kind of amazing. On the right, that's an experiment where what they did is, so this is normal senescence of the plant on the right. You can see it's a lot of cell death in the tissue there, a lot of browning. But the one on the left was genetically modified to overproduce cytokinins to keep those leaves young and healthy and fully functioning. Okay. Another hormone, now this is just a single hormone, abscisic acid. There's only one. So abscisic acid is often abbreviated ABA, and this one's famous. We call it the brake. Instead of the gas pedal, we call it the brake. And so it is commonly known to inhibit certain functions of the plant that other hormones would stimulate. So for example, I said that in order for seeds to germinate, you need gibberellins active in the seed. Well, before that, so this, when you got your seeds last week from the seed packets, they were dormant. And at that time, there was a fair bit of abscisic acid in those seeds. But as soon as you wet that seed, because you put it on a wet paper towel and then you encased it into a nice Ziploc bag to keep everything moist and wet, what you did there is you're diluting out the abscisic acid, you're wetting the seed starts to take up that water, that triggers gibberellic acid to get going. And hopefully when you look at your seeds tomorrow, you'll see the radicle coming out. At the very least, hopefully you'll see that your seed has plumped up, which means it took in water and it's getting busy to go ahead and germinate. So abscisic acid is keeping those seeds quiet until it gets all the signals. And so it kind of makes sense if you think about it from the plant's perspective, that when they make their fruits, they don't want the seeds to start germinating right away. Even though like fleshy fruit, it's pretty moist, right? You'd think, well, then how come seeds don't germinate and they're surrounded by all that fleshy, watery, fruit. They should germinate, but they don't. And it's because there's plenty of abscisic acid telling those seeds don't germinate yet. You're still in the fruit, so to speak. And so for the parent plant, what it wants to accomplish is get the fruit produced. The seeds are inside, but you don't want those seeds to start to germinate on the plant or even anywhere really near the plant. The mother plant, so to speak, wants those seeds to go elsewhere, land elsewhere, grow elsewhere, so they don't have to compete with their own young. And so very often you'll see that strategy. Not all plants do that, but many do. But regardless, you'll see the seeds, they stay dormant in that fleshy, watery fruit. And so they maintain the seed dormancy until all the signals have been met. And then bud dormancy too. So right now, the tree branches out here, if you look carefully, you'll see that there are leaf buds on the trees, but they're staying dormant. They are not sprouting into baby leaves yet, because of course that would not be a good strategy for these plants. We have a long winter yet ahead. And so there's plenty of abscisic acid in those leaf buds, telling the leaf buds stay dormant. Don't do anything yet. Another really important part of abscisic acid's role to play in the plant is to be able to respond and send the signal we're under water stress. And so when the plant is growing in some soil and the roots are like, uh-oh, I'm sensing a drop in soil moisture. Seems like the soil is dehydrating. That's a signal to them. We might be under a drought period here. This could be dangerous. So what happens is the abscisic acid will actually get produced in the roots in response to that soil drying out. And then the abscisic acid will course through the whole plant body. Do you know what these are? Stomata. They go all the way to the stomata. This hormone will go from the roots up all the way to the leaves, targets the stomata, and tells the stomata close down. So on the left, it's open. On the right, it's in the process of closing down. So the reason that that's happening is we're going to learn when water moves through the plant body. I mentioned it a couple times. It leaves, it exits the plant through the leaves, through the stomata. And so the plant is basically telling the stomata, shut down, conserve water, because I'm sensing with my roots that we're going into a drought period. So shut down. And so basically the plant is shut down at stomata and hope for rain. And if rain doesn't come, they're in trouble because now with their stomata shut down, they can't do photosynthesis because they're not taking in any carbon dioxide. So they're in trouble if they go through a long drought period. They're not going to grow very much because they won't be able to do photosynthesis. The other thing that will happen is the plant will say, shut your stomata, and then the roots will decide, so to speak, let's go find some water. So they will start to grow and forage for another patch of soil, hopefully nearby, that has some moisture, has some water that they can then take up in their roots. And so basically, the abscisic acid is doing several things. It's telling stomata shut down, but it's also telling the roots go ahead and grow. And so the shoot and leaves stop growing, but the roots continue to grow because they're searching for that very precious water. So it's lots of things happening in response to that soil drying out. Okay. Another hormone that they discovered quite a while ago is called ethylene. Again, it's a single chemical. Ethylene is a gas. So ethylene was the first gaseous hormone to be discovered, and it is produced in many different parts of the plant. Fruits, flowers, seeds, leaves, and roots. I mean, pretty much everywhere. And it can trigger its own production. So once ethylene gets made, it can kind of like cause this domino impact, make more, make more, make more. And ethylene should really be classified. Its nickname should be the stress hormone. That's really what it should be. Unfortunately, abscisic acid got that nickname before, but really ethylene should. Anytime you damage a plant, ethylene gets produced. So if you take a stem cutting of a plant, right where you cut it and damage those cells, it starts to give off ethylene. It's a way to signal I've been damaged. You know, it's important for the plant to be able to assess who's chewing on it or who's damaging it and try to figure out a way to defend itself. And so ethylene is part of that process. And so it is very, very important. So ethylene gets produced whether the plant is getting damaged by a deer chewing on it or a bunny rabbit or a fungus or an insect. Excuse me, ethylene will get produced if the plant goes through flooding or cold, when it's not supposed to be getting cold, and all of a sudden there's a cold shock or cold front moving through, ethylene will get produced. So ethylene gets produced under so many stressful conditions for the plant. It really should be called a stress hormone. And the other thing that it can do, which growers have taken advantage of, is it's used to ripen fruits. So ethylene is important in the natural process of getting that fruit to ripen up. And of course, this has been taken advantage of. So for example, tomato growers, tomatoes are fruit, technically. We'll learn more about fruits later, but tomatoes are fruit. But if you've ever eaten a tomato, you know how fragile they are. Can you imagine trying to ship tomatoes in a semi truck 2,000 miles and get it to its destination undamaged? It's hard to do. And so if they can ship the tomatoes unripe, so green, and then when they get to their destination, use ethylene to ripen up the tomatoes. That would be a bit safer. You shouldn't have as much loss due to damage in transport. And so things like that, they can use ethylene in order to maximize getting their fruits to ripen when they want them to, and hopefully having some nice fruits for us to buy at the store. It can also cause leaf abscission, which that's just a fancy way of saying that the leaves fall off the plant. And so abscission is a very specific process where the leaf stem or petiole will weaken in this very particular line of cells will start to weaken, and then that causes the whole leaf to fall off. So here they have the beaker with a plant in some water and not getting exposed to the gas. Here they have an apple. Apples, especially if you cut into an apple, it gives off a lot of ethylene. It's damaged, so it starts to give off ethylene. And then they put the beaker over it to capture the gas near the plant, and you can see that the leaves fell off.
Is that how Agent Orange works or no? Is that?
Yeah, defoliation. Mhm. Yep, that's causing leaf abscission. Yep. So what I just gave you a taste of are the 5 hormones. Yes.
So ethylene from one like species of plant can affect any other species of plant in the area also.
Actually, yeah, not all plants necessarily will respond to ethylene in the sense like with the ripening idea.
Mhm.
Some don't do that, but the leaf abscission idea is pretty across all the plant species, yes. Okay, so these are the 5 hormones that we call the classic hormones that we just walked through. And what I just did was give you just a little taste of what they do for the plant and maybe how we take advantage of that knowledge in food production. But there have been discoveries since the classic 5. There are other plant hormones. We just don't have time to get into all of them. We cover more in the plant phys class, okay? But okay, we got to stop there, and we'll get into a little bit more about how plants respond to their environment next time.